Yeah, so we'll talk about using persistent herbicides to control invasive plants at high latitudes in certified weed-free straw, and then some of the factors that go into persistence in our area with dealing with how absorption and desorption of the herbicide from those soils. And so, yeah, I'm with UAF in the Extension Service. I work at our Agriculture and Forestry Experiment Station as well. We're primarily based out of Anchorage and Fairbanks. And this project was funded through the USDA in one of those hatch capacity grant programs. So some quick background on herbicide persistence. If an herbicide is going to persist in the environment, it's evading some of the microbial degradation that can occur, primarily if an herbicide actually disappears from the environment. It's broken down by herbicides or the sun or water. The other way herbicides can end up leaving a site is through runoff or in plant material as they're taken up and plant material is removed from the site. We'll talk a little bit about that with some of the persistent herbicides, but runoff or leaching is one of the primary ways that an herbicide gets off site without prior to degradation. In Alaska and really in other places too, there's some desire to use persistent herbicides products to our advantage. When you're dealing with something that has a heavy seed bank, possibly has really early seed germination, or repeated germination throughout the growing season, having that herbicide there in the soil to control those seedlings can be to an advantage. Also, we deal with a lot, and I'm sure some of our other partner states in the West do as well, is remote locations where it's difficult to get to the spot once a year. And getting the logistics to get there makes using something with some persistence to your advantage. What we have to be aware, though, when we use persistent herbicides, that there is an increased chance of non-target damage from, especially if those things are active in the soil. Plants can still take them up, and new plants that are coming in, or adjacent plants, if there's some herbicide movement to laterally, can also end up taking up that herbicide and either show signs of damage or death. So what got us sparked in looking at herbicide persistence at high latitudes is aminopyrrolid and clopyrrolid's persistence. And it caught us off guard because both of these herbicides have properties that should show them move with water. So they should somehow leach off of site. And this, the KOCs here, that's the organic carbon coefficient of absorbance for herbicide, or how well that herbicide sticks to the soil and is corrected for organic carbon. These are taken from the herbicide handbook. And the key takeaway from this is that the larger the number, the more affinity that soil or that herbicide has to soil. And so glyphosate, you see, has 24,000 milliliters per gram. So by far and away the highest amount of affinity towards soil. And what that indicates, and that we know from in practice, is that glyphosate binds so strongly to the soil that it's not even able to get taken up by plants in most situations. Aminopyrrolid and clopyrrolid are far less, and chlorsulfuron is also far less, but a bit more than aminopyrrolid and clopyrrolid. Yet we've seen and observed persistence of these products in Alaska situations for several years at times, meaning the clopyrrolid and aminopyrrolid in the soil still being able to affect plants. So both of them, by their label, can aminopyrrolid and clopyrrolid can persist for a minimum of 18 months in the soil. But like I said, is evidenced in this picture of some potatoes that were planted in a field that had been treated several years prior with aminopyrrolid, we are seeing some damage, and primarily when you switch to using a sensitive crop species, something like potatoes potatoes die if you sneeze herbicide at them. So, but they do have great long-term weed control abilities, and we can use them to our advantage. We just have to figure out how to manage that persistence over the long term and still get that weed control. So the weed-free straw production in Alaska is a pretty big deal. The program is set. Our program runs under the NISMA standard, North American Invasive Species Management Association. We've been members there for probably 20 years now, but I guess not quite 20 years, 2006, if I look at my screen. It became a big deal for Alaska because of the Iditarod sled dog race. It runs through BLM lands. BLM wanted to require weed-free or local straw. The farmers who were already producing small grains up in the Delta Junction area jumped on the opportunity to certify their fields as weed-free, and they've been able to do so most years, and then provide the product to the Iditarod and get a premium price. On good years, you can find it at our local feed stores, and then also there's been a lot of interest in making wattles or erosion control tubes out of weed-free straw as well. And this is getting bigger than just in that one region of Alaska, Delta Junction. Potatoes are a big cash crop up here, and people are starting to use small grains and wheat in a lot of their rotations that involve potatoes, and hoping to keep certifying those as weed-free. So the weed-free straw and hay standards as well require inspection within 10 days of harvest. They're not supposed to have any of the prohibited weeds that are present. And then they also have to inspect storage facilities. Have a couple entries per field, get the whole perimeter, one entry point per 10 acres. And then all of that has to be harvested within 10 days. And they can have weeds present as long as there's no propagative parts in there. So none of the flowers or seed heads that are present in there, or stems that could be propagative as well. So producers in the Delta Junction area were running into some problems with certifying weed-free straw because of one species, narrowleaf hawksbeard, that's actually really sensitive to aminopyrrolid. We kind of wanted to study this because narrowleaf hawksbeard isn't super invasive in Alaska, but so it's been removed from that list. But it was kind of a canary in the coal mine, so to speak, of like, well, we have this weakness here. We have some things that could get into our fields that may be more invasive, say real hawkweeds, and others that have seeds with multiple germination and are difficult to control. And some of these herbicides might be really good at doing that. The farmers, though, have moved away from using things like aminopyrrolid or clopyrrolid because of the persistence issues. They just don't want to touch it. They don't want to take that risk that it's going to harm their potential to rotate to potatoes, or decrease their ability to move these products off site. So we wanted to start looking at their rotations and how we could help them get season-long control for their small grain and straw production. And typically they're rotating from 1 year fallow, and typically that's a chemical or plow fallowing to control weeds. Then they grow small grains and get both the grains and the straw off that, and then rotate into peas or potatoes after that. So Milestone and Transline and chlorsulfuron were the three herbicides we wanted to study in this. And the chlorsulfuron is kind of the status quo of, if you're going to do chemical fallow, they're doing that. They can also apply that to the field after they've planted, and everything's still short when they are in the small grain portion of their production. Milestone and Transline both have some interesting restrictions on what you can do with the product. If you're growing hay and you apply a milestone, it needs to not be moved off site for 18 months. Translines within the same season. You can't have any of that material get into a compost facility for at least 18 months. And you're not really supposed to plant another crop into it without first conducting a bioassay on those crop species. There, interestingly, isn't really a lot of restrictions there for straw production. It kind of falls into a different category, I guess, of where it can be moved. But the reason they have these restrictions is because aminopyrrolid and clopyrrolid, obviously you're using for weed control in these small grains or in hay. They don't damage that, but they are taken up by those plants. And as they're taken up by the plants, they're not broken down or changed. They remain aminopyrrolid and or clopyrrolid. If, as this diagram shows, if you then feed that crop, hay or straw, to cattle, horses, or other animals, it remains in its parent form, clopyrrolid or aminopyrrolid, as it passes through the animal, gets into the manure, and then can potentially damage plants, other non-target species, if it gets in the compost, or if it goes into, is used as a mulch in vegetable production, etc. And this is something that the compost industry in North America runs into every now and again, where they have basically hot piles of compost that are hot with an herbicide, not necessarily hot with the heat to break things down. And they're not seeing these herbicides actually break down in the composting process either. So what we wanted to do was kind of toy with the rotation period a little bit and see if we, and also study kind of how that herbicide changes over the time with these rotations. So our first year was in 2020, and we did a fallow year. We applied our herbicides in the spring of 2020. 2021, we collected soil samples and for both analytical determination of herbicide presence as well as bioassays, and then planted barley in that year. We repeated that same process in 2022, and now we're heading into 2023, where we're going to be planting peas, Austrian field peas. It's our first sensitive species in this crop rotation pattern, and so we'll see what happens. We had a heavy snow year, so our fields are still drying, and we haven't had a chance to get out and do that work yet, but it is pending. And then finally in 2024, we're going to rotate to potatoes. Potatoes probably the bigger cash crop of these in the rotation, and we'll see what happens. Our bioassays that we do are done with narrowleaf hawksbeard or Crepus tectorum, the same species I mentioned, kind of was making it hard for people to certify their fields as weed-free. That species is really common and easy to collect in the state. It's also very sensitive to aminopyrrolid. We have in the past told folks that we think you are safe to plant potatoes in a field after applying milestone if the narrowleaf hawksbeard comes back. So we also planned, besides looking at the herbicide in the soil, to look at that quantity of herbicide that's actually taken up in the straw, because obviously movement of that herbicide off-site could be a problem. You'd think it wouldn't be that big of a deal for straw wattles or erosion control tubes or dog bedding. However, sometimes I've heard through the grapevine that, at least with the Iditarod, the dog bedding sometimes gets picked up and put into people's compost piles afterwards. So we wouldn't really want to see that happen. So this chart shows you what our certification success was in both 2021 and 2022 for each one of the herbicide active ingredients. We had 5 plots for each treatment in each area. So one plot or one area is in Palmer, that's in South Central Alaska. The other's in Fairbanks, which is in the interior Alaska. They both have similar growing seasons. Palmer's a bit more maritime. Fairbanks is more interior, so continental. A little bit warmer summers, but otherwise the seasons are about the same length and the like. And so what you can gather from this is that we're, at least in the Palmer area, we're getting quite a bit more certification out of the aminopyrrolid treated plots than we are with any of the other herbicides. All the herbicides tend to perform a little bit better than the control treatments, as you would expect. Fairbanks was a little bit different than in the sense that it didn't really perform that much better at at getting fields or plots certifiable. And so one of the reasons I think that might be is that in Fairbanks, we had quite a bit of predation by cranes early in the year. One of my colleagues had a graduate student that was chasing them away from their plots, and I think they chased them into mine. And so we had some really thin grain production in the Fairbanks area, and so that might have decreased some of the competition we would have got from the plants themselves, the crops themselves. But overall, the aminopyrrolids seem to perform quite a bit better than the other herbicides at getting these plots certifiable as weed-free. And then the percent cover of the weeds that were present in those plots also gives us the same indication. And this is looking at just prohibited weeds, and those would have been prohibited weeds on the NISMA and Alaska lists for certifiable weed-free straw. And then there's two pictures here. The one in the upper right is one of our control plots in Palmer, and you can see all that green in there is hemp nettle and lamb's quarters and various other weed species that were able to grow up and compete with that crop as well. We can see this a bit in the dry weight that we were able to get off of the plants, although nothing really differed statistically. You can see really starkly, though, the decrease in the amount of mass of product of straw we were able to get in our Palmer or Fairbanks plots because of those cranes. They really did a number on our seeding. But aminopyrrolid plots seem to be just a little better, but they're really not statistically any different than our other plots as far as actual production goes. This is a list of our prohibited weeds that we found in these plots with scientific names: narrowleaf hawksbeard up at the top, hemp nettle, one of our knotweeds, and various others that are on there. Aminopyrrolid obviously tended to have quite a few less of them, so just kind of an interesting thing to look at some of the weeds that we were dealing with at these two locations. So going back to herbicide presence in the soil and looking at both years, is that with our bioassays. Our bioassays were conducted for 3 weeks. We planted 10 seeds of that Crepus tectorum in each of the Petri dishes with soil from the collected sites. And so you can see an example of those Petri dishes from, I'm not sure which area is actually shown in the picture, but you can probably make a guess as to which one had the aminopyrrolid in it. What we're seeing here so far is that in 2021, the aminopyrrolid controlled the Crepus more than any of the other treatments, except that in 2022, it did not do so as much in Fairbanks. It did, though, in Palmer. There's some possible reasons for that. In Palmer, our management practice for those fields is to leave them in grass until we are ready to do an experiment. and then they till up that grass. And so there's quite a bit of an organic matter content that's in there that could be keeping that herbicide in the soil. In Fairbanks, the management practice presently is quite a bit different. The whole field area that we would do research in are plowed regularly for weed control, and it's pretty much just broken up soil with very little organic matter in it. It's kind of what you would think is poor soil management, especially if the area was windy. It's not particularly windy there very often, but we do see quite a bit more of the herbicide sticking around with aminopyrrolid. The other herbicides don't necessarily differ from water that much, save clopyrrolid in 2021. Although 2022, the herbicide seems to have possibly gone away. Now, we are redoing our bioassays for 2022. I had some student research technicians last summer that were really ambitious, and in the growth chamber that we were using, they raised the shelf up to get closer to the lights, and this was done after we did the 2020 after we did the 2021 bioassays. So we're redoing the 2021 bioassays at that same height. They'll be done actually this week. I don't anticipate that anything's that different because at 3 weeks, most of these plants are still kind of going off of a lot of the what's in the seeds mostly, and just getting up. But we'll see. So we can't say definitively that it looks like the herbicide is going away with our 2022 soils, but it seems as though it might from this data. Oh, I skipped over. I left a slide out about detections. We were able to detect the herbicide in the 2021 soils, but we're still working on that for 2022 as far as analytical detection. We did this analytical detections with an LCMS triple quad, so liquid chromatographer with mass spec, so that we're able to get a pretty good, I believe, a 1 part per billion detection limit. So we want to really kind of try to understand the processes of absorption and desorption. So absorption is where that herbicide sticks to the soil. Desorption is when, after an herbicide has bound to the soil, how easily does it come off? And in our Alaska soils. And so wanting to compare Fairbanks and Palmer soils to see if we have soil characteristics that affect that absorption. And what we ended up deciding to do was vary our pH. And then we also wanted to see if that absorption behavior actually explains what we're seeing at our field sites. Now, to back up to pH, the reason we chose pH was because when you run these absorption experiments, you do a lot of preliminary work to see, how long does it take for the herbicide to stick? At what temperature do we need to have it at, et cetera. And what we found was we weren't really able to get it to stick at just without varying that pH. So I'll get into that here in just a second. But the basic background of this is that when you do absorption experiment, you equilibrate your soil with clean water, and we usually put a calcium chloride in the water as well. Then you spike that soil water solution with a given amount of herbicide. We then agitated it for 24 hours. That's just to equilibrate it again, get that herbicide to contact soil in the soil water slurry. We then take some of that water out, and so we use that water to figure out how much herbicide is still in it. What's not remaining in that amount of water, we just assume has then been absorbed to the soil. We then refill it with fresh water, and then we agitate it again, and then take that soil sample again. And so that soil sample that's taken that second, or the water sample, sorry, that's taken the second time, that was after refilling it with fresh water, tells us how much herbicide then desorbed from from that soil. All of these are gone through a solid phase extraction and cleanup procedure to prepare it for running through our LCMS mass spec. We varied our pHs in this all the way down to 2, which is super low. 3.5 is actually some of the 3 to 3.5 is some of the low end that you could find in Alaska. We have pretty low pH soils because of the high organic matter content and cold soils that are pretty darn new. And then 5 was kind of the native soil pH that we had. And then we had the 2 from the interior and south central as well. And so from all of that experiments, we're able to calculate the KD and KOC. KD is a absorption coefficient that gets calculated from these. And then KOC is that same absorption coefficient, but corrected for the organic matter content of the soil. So it's basically the concentration of the herbicide that's in the soil over the concentration of the herbicide in the water. And then the KOC is then that number divided by the fraction of organic carbon. What you can take away from this is that the bigger numbers indicate higher affinity of the herbicide toward the soil. And so we see that as the pH goes down, the herbicide affinity towards the soil increases, and that's statistically significant increase in that absorption to the soil. We're getting almost no absorption in the pH 5s, but quite a bit in 3.5, and then a lot in pH 2. The reason we think that might be occurring is because at those higher pHs, the amine group on the amino pyrrolid is likely to be completely deprotonated, meaning it loses those hydrogen ions, and then so it doesn't have the right charge then to stick to some of our soil and clay particles that are in there. So this year we're looking at planting the field peas as our next step in future work. And then we need to look at what's driving persistence overall, like in our future. It's not part of this current grant, but we're thinking of the next steps. Why is what we're seeing in these field applications where we're seeing herbicide persisting in the environment at farms for many years and sticking around in the soil for at least a year or more in our experiments. Why does that not necessarily reflected at that pH 5 native pH soils in our soil isotherms? Some possible reasons is application to dry soils. We don't, in those isotherm experiments, you're doing a soil water slurry, and that soil water slurry is a 1-to-1 solution. And so if you think about that, that's like being in a really silty, dirty river. Up here, we're really used to that with our glacial fed streams. You go rafting and you can hear the silt running against, it sounds like sandpaper running against wood on the bottom of your boat. But you might notice the same thing in a big picture, a big muddy river. Those still probably aren't quite a 1-to-1 solution. There's less soil in it than that, but we're talking a lot of water in there compared to when we apply something to a dry soil, and then there's a lot of time for hysteresis, basically changing of the soil particles as they bind to those the herbicide and possibly tie it up a bit more. There's also a big role that plant and animals uptake and recycling might play into this, where folks have seen problems in Alaska in an agricultural setting with persistence and then rotating to these sensitive species has really been in hay production as opposed to in straw production. What happened in hay production is keeping people from doing it in small grains and straw and taking that risk, but it was in hay pasture situations. when you're in a hay or pasture situation, you're not removing that entire plant material that has the aminopyrrolid or clopyrrolid in it. Instead, that plant material is able to take it up and then degrade back into the soil when it's not moved off site. And so that could play a significant role in these things persisting in these northern environments for many years beyond what we would necessarily expect. We're also interested in conducting this summer, looking at biochar and wood ash amendments and seeing if we can actually start to work to tie up that herbicide in the soil with biochar or wood ash, because those are materials that are known to add quite a bit of stable, meaning thing won't be broken down, organic carbon to the soil. And so if we add organic carbon to the soil and get quite a bit more binding sites, there's potential that the herbicide will be bound so strong that it will no longer be available to plants. There's also potential that you could increase the persistence if it somehow still remains available to plants, or you might mess up your ability to use herbicide later, because if you have an absorbent, a whole bunch of an absorbent material in there, and you need to use that aminopyrrolid now, but it's going to get absorbed so strongly to the soil that it's no longer available for plants in its persistent form, that could also be a potential factor. How are we doing for time? Because I could take some questions about this work now, and then I also have some other stuff on basal bark treatments if we want to get into that.
Yeah, we're good on time. It's 12:38, so we have about 20 minutes left. So if folks want to go ahead and pose a question now about what Gino has presented, you can type your question in the chat, or you can also just take your mic off mute and ask your question. I do have a question about this work, but I'll let other folks get a shot in before I ask mine.
I want to do that now because the other stuff's a bit different, but along the same lines because we use the same chemicals.
Yeah. So I'll just go ahead and pose mine. Obviously in integrated pest management, we're always trying to balance risks and benefits associated with the pests and the pest management. And, kind of the risks of using like something like aminopyrrolid, or the persistence in soil and the persistence in the plant materials. So it seems to me like in a situation where you're trying to produce straw that has to be certified, the benefits of using the aminopyrrolid outweigh the risks, in the sense that if it's in the straw, maybe that's not as big of a problem depending on how the straw is going to be used. But I suppose in a pasture situation where you've got animals grazing that material, in that case, I'm guessing the risks probably outweigh the benefits, because obviously you're now worried about that getting off into compost, which we know from the EPA's sort of it's fall 2021 sort of decision on mitigation procedures. That's one of the big things I was worried about. So I was wondering if does that sound right, or is there something I'm not thinking about?
Oh no, that is right on with it. I think that the risk and we need to get better information about how long it can actually persist in a pasture environment. Presently it's like an 18-month standard for that. And then grazing animals is actually, I can't remember as far as when you can start using the manure from those sites, if it was still 18 months or if it's needs to be, or if it's, because they just address the 3 or 4 days after that the animal's been eating a clean environment. And so those are the things that I think where we need more study to really evaluate the risk in that setting. And primarily because the compost industry is still getting hits. And so either it's because we don't know that it's still there, or it's because there's been some lapse in some of the reading of the label and following those label instructions.
Yeah. Yeah, that actually leads to the second question I have, which is, there's a variety of mitigation procedures, like you said, pulling the livestock off for 3 to 4 days and feeding them on, non-treated forage as a way of mitigating that problem. It sounds like maybe that mitigation strategy works sometimes, or it may be an issue of people complying with the mitigation.
I imagine that in the realm of manure management and manure being moved off site, that's probably people not following that, because that's much easier to do. In the area of whether or not the field, a hay field or pasture, still has aminopyrrolid cycling through it, 2 years out. That's kind of what's unknown. We've certainly seen it in Alaska several years post application in pasture fields, when people rotated to sensitive species. So that's the wild card, and it may be that in a lot of the places where pHs are much higher than what we have, more neutral, ours tend to be pretty acidic, that's really not an issue. And that really the issue tends to be once we start getting the acidity lowered, or pH lowered, acidity increased, and now we're getting more absorbance of that herbicide.
Yep, makes sense. So I'm not seeing any additional questions in the chat, and I don't see anybody with their hand raised. So if you want to go ahead and take it off into basal bark treatments, let's go ahead and do that.
Yeah, so we have also used aminopyrrolid quite a bit for managing Prunus patus. It's a chokecherry tree that's invasive in the state of Alaska. And I kind of wanted to give you all an update on that. We did print in Weed Science this last November about herbicides exuding from the roots of treated trees from following basal bark treatments. And so we're really interested in treatments that are designed to limit the amount of herbicide getting into the environment, so these direct treatments. We picked on basal bark treatments because aminopyrrolid's labeled for that. There are other treatments like cut stumps and frill treatments and injections that also are direct treatments. Our applicators, though, that we've talked to, typically we don't see any non-target damage from those, but they were seeing some kind of dead zones around trees from basal bark treatments. So real quick, to kind of go over that, we had done these basal bark treatments, and we found after treating individual trees, and then we'd move out and treat a different individual tree, we're able to find aminopyrrolid in the soil about a year after those treatments. It was typically below our limit of quantification, so we could tell it was there, but we couldn't tell you how much was there. We could just, with an instrument, would say, yep, I see it, but it's too much of an error to differentiate that amount from the low end that we can really detect. We had done treatments with aminopyrrolid and triclopyr. We couldn't find triclopyr in the soil, just the aminopyrrolid was present in the soil. And we did also see on-target damage in the areas around these trees that were treated. We looked within what's called the drip line, or basically as far as the branches can go. It's a way arborists usually use to define the area that where most of the tree roots are. And this first round of data can all be found in that paper that I recently published. We also saw the data from our bioassays showed us pretty much the same thing. All our herbicide treatments were different than our controls. We ran one control with just basal oil, and no herbicide in it. But none of the herbicides actually differed from each other. Triclopyr had slightly better growth, but not so much so that we could actually tell a difference from it and aminopyrrolid. So what we wanted to do after this paper was actually start to look at what happens if you actually treat all the trees in a plot. Because we wanted to focus on with that first paper, looking at root exudation, and could we figure out if the herbicide was coming out of the roots. And we surmised that yes, it did. We actually had a lab experiment as well where we controlled for it running down the stem, and we're still able to detect the herbicide in the soil and got the same results from the bioassays. But we wanted to see if when you start to treat an entire plot, do we then basically compound that? Because now we have trees that are sharing their root zones and getting treated right next to each other, rather than things that are spread further out. And then also wanted to see if we could help out by reducing the rates, see if that then keeps us from being able to, or keeps the herbicide from being able to damage other trees, shrubs, and plants in the area. So we did our treatments in 2021. We treated all trees greater than 2/3 of a meter in height, and they had to be, thick enough around to be able to fit into that label recommendation of being able to treat for herbicide or with basal bark treatment. And so that's like I think 6 inches is the max, and whatever you can squirt is the smallest. And we use the full and half label rates of Garlon, which is a triclopyr product, Milestone, and then had an untreated control. We mixed them all in a paraffin oil, basal oil blue. And this is the picture, shows you just kind of where we were working. There's a couple of people back there. Those are probably choke cherry trees with some alders mixed in, and just kind of a thick jungle of stuff to climb through. So we looked at percent defoliation of the trees from these treatments that we did, and it was interesting to see that Milestone at its full rate performed better than all of the others, but the Garlon at both its full and half rate were virtually identical as far as how much defoliation they had 1 year after treatment. Milestone at a half rate was not significantly different from them, but had slightly more defoliation. And so the thing to keep in mind with this, though, is that basal bark treatments take a long time to work. You basically get the herbicide to stick to the trunk of the tree with an oil fin, with an oil carrier that helps it stick, and then absorbs through the bark. And so sometimes you'll see partial defoliation 1 year after treatment, and then the tree's not fully dead until the second year. So we're planning to get out there again this summer and take a look. My hunch is that things will catch up with some of these. I don't know or think necessarily that they will catch up to the full rate of Milestone, but they may be at acceptable rates. And it certainly looked like they were probably at acceptable kill rates when we were out tromping around in these plots. So using ratcheting back your amount of herbicide could save you some money, as well as putting out less herbicide in the environment. We did see non-target damage. These are pictures of what we were looking for. That upper left is an alder tree with cupping of leaves, characteristic of auxin herbicide damage. The one on the upper right is a mountain ash. Down below that, I believe, is a false spirea, and then a spruce tree as well on the bottom left. Spruce trees are really sensitive to aminopyrrolid, so you're never really surprised to see that, but still, when you're doing that direct treatment, you don't necessarily expect it. This time we didn't correct for root to root contact or anything like that, but either getting out through the soil environment or root to root contact. So this chart shows you the non-target damage that we observe by treatment. And so what you see here is we have total plants with damage that we saw in each of those treatments, and Milestone by far and away took the lion's share of the total plants that we had saw damage to for many of these treatments. Plots, though, we really only saw it in 4 plots. We had 10 plots total, 5 in each area. This was in Fairbanks and Palmer again. A lot less with the milestone half rate. And then Garlon, we only had one plant that we ever saw. That plot happened to have the highest amount of herbicide put into it of any of our plots. Just had the densest number of trees, or they were likely also bigger. We calculate herbicide amount based on the basal diameter, and then we spray up the tree to about 18 inches, or the height of my extra tough boot. We're able to calculate the amount of herbicide that way based on the volume of a cone and how much herbicide we used over the whole plot. So the drawing your attention over to the next 2 columns of number of treated trees treated in the plots, and then volume, or in milliliters of herbicides in the plots that had the damage. What this shows you is that it really varies quite a bit. So we're seeing damage not necessarily because we're using more herbicide, or because we're treating more trees. We're seeing damage over that wide array of those. We were only able to actually detect herbicide in the soil in 3 instances, and those were all in the milestone full rate plots. So I don't have the bioassay data for this one yet, but looking at these plots using bioassays, observations of non-target damage, and detection and analytical detection is really going to be your best way at assessing that herbicide persistence. And I say this because with the soil samples, we were only able to find herbicide in 3 of those plots, yet we saw damage in at least 4 and more in the milestone half. So the herbicide's there, it's doing something a year later, but we didn't necessarily were able to find it. But you can use reduced rates, and that has a really good chance of lowering the amount of non-target damage you see. You can also use herbicides that are less persistent, and that seems to help. We found that in the first study as well as in this one. Using Garlon seemed to be able to reduce a lot of that non-target damage and the persistence. I would say, though, that the non-target damage we observe is really slight. And in the past, when I've gone to visit some of the spruce trees that we saw it on, it doesn't persist into the tree. It's not killing off of these trees necessarily. It's harder to track smaller herbaceous plants, though, and to make sure that they're not dying later on. So yeah, you can try to avoid non-target damage by using really careful application, herbicide choice, and hopefully ratcheting back that the rate. And we're going to be visiting these sites again this year to kind of reconfirm some of this stuff and see how things are going.
Great. Thanks, Gino. That was terrific. Again, everyone, if you have a question either about the basal bark treatment or the previous section of the talk, which talked about production of weed-free straw or aminopyralid or clopyralid use on pastures, you can go ahead and just type your question in the chat, or you can raise your hand, or if you want, you can just take your mic right off mute and ask your question that way. Gino, I have just a quick question. How thick, or sort of, once choke cherry gets in there, are the stands pretty thick and there's nothing else growing in there, or is they pretty wide? I guess it probably depends, right?
They tend to, especially when they come in thick with small diameter trees, they can grow in and seem to keep a lot of things from growing underneath them. We lose dogwoods, currants, cranberries, ferns, and all sorts of things in those situations. They tend, as the trees get bigger, there's some theory that maybe they start to thin out. We're not necessarily always seeing that, though. I think that when we do, it's possibly that the bigger trees are starting to shade out some of the little ones, and we're just not getting as many little ones coming up. But yeah, they grow dense, and even when you read about them in other species of chokecherry and Forest Service publications, et cetera, they'll point out that because of the suckering and most of the cherries falling pretty close to the tree, whether it's birds eating them or just them falling down, tend to form pretty dense thickets at times.
Yeah, makes sense. Okay, I'm not seeing any questions in the chat or any additional questions in the chat, or I don't see anybody taking their mic off mute. So I just want to thank you again, Gino, for a great presentation. This presentation has been recorded, so if anybody wants to check it out, it will be available, probably not today, but when Steve gets back from vacation on the YouTube channel. We won't have an IPM hour in June. We'll be up visiting you in Alaska, Gino. Should be fun. But we will have a IPM hour in July. That's scheduled for July 12th, and that will be Jill Silver talking about lessons learned. Excuse me. Lesson learned from 20 years of management of invasive knotweed on an unregulated western river. So thanks again, Gino.
What was her name again?
Jill Silver. She's she's up in Washington at the 10,000 Years Institute.
Great. We're looking for some knotweed people.
Very good. If you want to find out more, you can actually go to the website, and it's got a listing. If you look under the About tab, and it's got publications, the IPM hour is listed there, and it's got a listing of all the IPM hours up until October.
Great.
Thanks everyone for joining us. Enjoy the rest of your day.
Thanks.